Okay, I think it's recording. All right, so way back to the beginning of the year, what is psychology? Hopefully you know that by now. Study of behavior and mental processes. It's considered a science. Our father of psychology is Wilhelm Wundt because he established the first psych lab. We talked about introspection, which is that deep self-analysis. We did introspection with Hershey Kisses at the beginning of the year. G. Stanley Hall established the first psych lab. We said think of the Hall of Psychology. Wilhelm Wundt, Hall was one of his one of the people who came after him. Structuralism and functionalism were the first two early schools of psychology. Structuralism focused on the basic elements of the mind, about what we think, and then functionalism focused on why we think what we think, and that was William James studied the purpose of what you're thinking about. Early women, we talked about Mary Woodin Calkins, should have been the first woman to get a PhD, but Harvard refused her, but instead she was the first woman president of the APA. Margaret Floyd Washburn did get that PhD, and then Dorothea Dix, we've talked about her for the humane treatment of people in mental asylums. Structuralism, that's a repeat. Some of these slides are duplicates, actually, by accident. All right, next group of people, John B. Watson was the founder of behaviorism. We know him for the little Albert experiment. And this is where the nature versus nurture debate popped up. Nature, remember, means anything determined by genetic inheritance. Nurture means the environment and experience. Nurture can include women who are pregnant and how the environment is affecting babies that are in the womb. That can also count as nurturing. B.F. Skinner, we know for behaviorism, and he did the rewards and punishments. We talked about important people last week, but this is just the intro stuff. Carl Rogers, Abraham Maslow, humanism, freedom, and personal growth. Cognitive psychology, remember cognition is anything having to do with mental processes and thinking. Some of the big cognitive psychology people were Jean Piaget, Noam Chomsky, and Theodore Simon, but main one we talked about was Piaget. Biological perspective looks at physiological processes. Sperry was the left and right brain, split brain experiments. Evolutionary psychology looks at natural selection and how different characteristics help give people a survival advantage. David Buss studied relationships and natural selection and mating, what we look for in mating preferences. Ethnocentrism is when we tend to look at our own culture as the standard. A lot of early psychology was studied by upper-class white males, so that was kind of biased, but they're trying to redo lots of studies now. I didn't put all the different branches of psychology in here because I felt like that stuff was pretty easy, but I did want to remind you that industrial and organizational psychology studies ways to boost production and improve working conditions. So that's a psych degree that does not treat any kind of patients. It's applying psych to the workplace. The APA, the American Psychological Association, was founded by G. Stanley Hall with Mary Woodin Calkins as the first president. All right, the next part of Unit 1 was all about research. Research is a huge part of the exam. We know for sure that one of the FRQs is going to be about research, where they're going to describe some kind of research. They'll ask you, what kind is this? What are the pros and cons of this? So if you go back and study a section, I would study some of these slides here. So we have observational or descriptive research, correlational, or experiments where we can manipulate variables. The correlation and descriptive ones, some examples of that are naturalistic observations, case studies, surveys, and quasi-experiments. The problem with correlational and observational studies is you cannot manipulate any variables, so you can't determine causation. Naturalistic observation, remember, you cannot let the subjects know they're being observed. Let's see, we'll skip some of that. Case study, you're getting a lot of information. We talked about case studies on serial killers, case studies on rare diseases. The problem is you can't generalize that information across the larger populations. Case studies, usually good for new and complex information, but again, you can't generalize. Survey, they're really good for gathering lots of data, but it's self-reported and sometimes people lie. Cannot imply causation. Surveys can be part of the operational definitions within an experiment, though. Why is the screen like wasting all this top and bottom part? Like, see all that black space on the screen? I don't know why it's like that. All right. Surveys are good for gathering lots of data, but they're vulnerable to different biases. Social desirability bias. Remember, that's where you answer in a way that makes you sound better. Like, sure, I give lots of money to charity. Sampling bias is where you only give the survey to some people, so that could skew the results. And then a response set, that's when people are kind of lazy and they hit agree, agree, agree. They don't like really reply to the survey. Problems with just how you word it. Let's see, different types of studies, longitudinal versus cross-sectional. Longitudinal, you study the same people over different intervals over an extended period of time. You can see really good change over time, but it's time-consuming and expensive, and people might leave the study. Cross-sectional is better in that it's quick. You're going to study different age groups, but all at a single given time. So it's less time-consuming, but you have a lot of different people, so that leaves a lot of different extraneous variables. Descriptive statistics, when we're looking for correlations, we can measure a correlation by looking at the strength of the correlation. It's only going to be between 0 and 1, can be positive or negative. Remember, a positive correlation means two variables increase together. Negative, one increases while the other decreases. We can measure that correlation with the correlation coefficient of R. R equals 0 to 1 or 0 to negative 1. So remember, positive, they both go up together, they go down. Negative, one goes up while one goes down. We can also use the number line to look to see if it's a low, moderate, or high correlation. Scatter plots are a way to graph that. Positive correlations, there will be a line sloping upwards. Negative correlation, a line sloping downwards. No correlation, they're all over the place. More often, they're scattered, but they generally move up or they generally move down. Correlation, remember, correlation cannot determine causation, but it helps you to predict something. So the higher the correlation or the stronger the correlation coefficient, the more you can predict it. But sometimes things are correlated, but again, they're not causally related. Like foot size and vocabulary are positively correlated. But there's a third factor. So they call this, and this keeps showing up on the AP test, the third variable problem, that there's often another variable that's really the reason. So foot size and vocabulary are positively correlated, but really it's also because their age. As they get older, their foot size gets bigger, and that helps, and they have more years of school and their vocabulary goes up. So the third variable is just talking about when there's correlating variables, there's often another one that's changing it. 0.78 is moderately strong. 0.05 is weak positive. Negative 0.43 is moderately weak negative. Negative 0.92 is very strong, but it's a negative correlation. All right, experiments. Remember that experiments are important because that's the one way that we can determine cause and effect. But you have to set it up to where you're manipulating only one variable. We have two variables, the independent variable, that's what we're manipulating, and the dependent variable, which is what we're measuring or is manipulated. How does X affect Y? Whenever we're trying to measure something, we have to use an operational definition. How are we going to measure this? How can we quantify and clarify what is meant by each variable? They really like to ask that on research questions. What was the operational definition? How did they measure happiness? How did they measure success? How did they measure academic achievement? So for example, if we say studying helps students succeed in school, we could say studying is the independent variable. The operational definition is the number of hours. Success in school is their grade on a specific test. So we got to be more specific. We have different groups. We have the experimental and control groups. The experimental group gets the independent variable. The control group gets nothing, or they get a placebo. That was in, if you do the practice FRQ on the mock exam. Control group was one of the terms on there. Random assignment, that'll be on the next slide here, or one of the ones coming up. Extraneous variables are any other variables that show up that could influence your independent variable. You try to catch them ahead of time, and then they become controlled variables. If you don't catch an extraneous variable, it becomes a confounding variable, and that means you didn't catch it ahead of time. It interfered with the experiment. When a potentially confounded variable is controlled, the logic remains intact. Nuisance variables are another type of extraneous variables. They're things you can't control, like you're taking the AP test and a fire drill happens, or if somebody pulls the fire alarm. Hopefully that won't happen, but it could happen, and that could affect your score. That would be a nuisance variable. And you'd probably get a retest. You'd have to take the retest. Placebos are what we give to the control group, so that when you're testing out a medication, we know, is it their actual, just their beliefs that they're getting better, or is it the actual independent variable of the medication? The placebo effect is when a person has a change in their behavior because of their expectations. To avoid the placebo effect, we use the double-blind study. So in a single-blind study, the patients don't know what they're getting. So single-blind and double-blinds help with the placebo effect. Double-blind, both the patient and the researcher do not know who is in the experimental group and who is in the control group, and they use assistants to help them, and that's how they do that. Populations versus samples. So this can be for both surveys, but it could also be with how you get your groups in an experiment. Population is anybody who could potentially be part of the study. The sample is when you just get a portion of them. Random sample is when you randomly select people, like drawing names out of a hat or using computer-generated random names. Representative sample is when you specifically pick groups that are a smaller portion that represents the overall population. So if the population is 20% Hispanic, then 20% of your participants would also be Hispanic. Random assignment, once you get your sample, you randomly assign them to the experimental or control groups. So let's see. So random sample, you get the people. Random assignment, once you have them, then they flip a coin, they draw the name out of a hat, and you randomly assign them to a control group or experimental group. They like to ask this on the AP test. When you do an experiment, you have to have random assignment or your study is flawed. So that way we know that they were randomly put into the experimental or the control group. They like to put that on FRQs. Like, what was wrong with this experiment? They didn't use random assignment. Like if I said, I'm going to put all my first period kids in the experimental group and all my second period kids in the control group, that's not random. That would make my experiment flawed. Advantages and disadvantages. The big advantage is we get cause and effect. The disadvantage, it's really hard to create a good, fair experiment. It's artificial, and there's a lot of ethical and practical issues. Biases, the hindsight bias. We call this I knew it all along phenomenon. This is your tendency that once you know the result of something, like when you're making your test corrections, you're like, oh, I knew that, but did you? You got it wrong. You probably didn't. That's the hindsight bias. Confirmation bias is when we're only looking for and interpreting information in a way that confirms your beliefs, like always watching a news channel that runs articles that support your beliefs. Sampling bias, again, when you only give your survey to people who would answer it a certain way. Statistics, we have a little bit of that, and there's a chance there could be a little bit of math on the test. If you took the mock exam, there was one or two questions where you had to do a little bit of math. But the main ways you would do that would be with measures of central tendency. The measures of central tendency are mean, median, and mode. If you have to do any math, it would be calculating a mean, and it will be something you can do without using a calculator. So mean, remember you have to add up all the numbers and then divide by the quantity of numbers that you had, and that's your mean or your average. Median, remember that you need to sort them into numerical order and find the middle number. If you have an even number, you're going to subtract or take the average. So add those two and then divide by 2. And then mode, you're going to again put them in numerical order, and you're going to see which one has the most. So this one has 4 13s, so that's the mode. If there were 4 14s also, we would call that bimodal. It means there's 2 modes. I don't know if there's such thing as trimodal, maybe. All right, measures of central tendency. Another thing that they could ask you about is standard deviation. That's how much the scores vary from the mean. How high are they from the average? High variability is when the scores are far apart from the average. So we had those two streets where people were driving fast and slow. On Perfection Boulevard, the standard deviation is low, 2.87, and that's because all these speeds were close together. On Wild Street, their standard deviation is higher, and that's because the numbers are farther apart here. So they still had the same mean, but the numbers varied a lot more on the one with the higher standard deviation. So you don't have to calculate standard deviation, you just need to recognize it. This is a normal bell curve showing how many scores are closer or farther from the mean. I don't force you to memorize the percentages on there. If you took stats, maybe you know them. Some people memorize those or force their kids. I want you to learn a million other things instead. All right. Remember that statistical significance means that the probability that the data was just because of chance is less than 5%. Let's see. Meta-analysis is when you combine lots of studies together and look for trends. It's important to replicate studies to see if you get the same results. Ethics, deception, animal research, you can allow deception, but you got to debrief. You've got to get informed consent. You can't coerce people. You got to let them withdraw. Do no harm. Even psychological harm is often prohibited. It's important to debrief. Go back and correct any misunderstandings. Subjects have a right to privacy. Animal research, you need to justify anything harmful you're doing to the animals. And then get approval from the research group. They also call them IRBs. That's an independent review board. All right, that is all of that unit. Where is my other unit? Unit 2. All right, Unit 2 was the brain unit. Where are you? Why aren't you opening?